Yeah, here we should start. So, good morning, everyone. Thank you for coming today. Also for the people who are watching us online. Just before we start, I want to make a little announcement. You probably have received an email, but even if you haven't, next week we have the Louis Thaler conference. That's the big conference of the SEM seminar series of this year. So it's next Friday at 11:30. It's not at the in this room, in the Talera room, but it will be in the amphitheater of the CNRS, at the delegation of the CNRS at Woodement up the road. So again, it will be, of course, on Zoom. We have as guest Anna-Lisa Line from the University of Helsinki. And for the people who are here next week, Annalisa is coming and she's staying from Thursday to Friday. So on Thursday, there's a possibility for the permanent and for the non-permanent, for the students to meet with her. So if you do wish to meet with her and chat before her talk on Thursday, send an email to the organization of the SEM at SEM, sorry, and we will put you in contact. Again, leave your comments for our speaker on the Q&A, and I'll give the floor to Ignacio Bravo for introducing our speaker.
Thanks a lot. Well, it's a pleasure to have Sandra Cortijo presenting at the SEM. We met 1 year ago at the conference on noise in biology in Tübingen. And I think she will then today present developing on individual variability. So Sandra did her PhD in Paris on quantitative genetics of methylation, and then two postdocs in Cambridge in a plant laboratory, working first on chromatin integrity and interactions between chromatin and transcription factors, and then on interindividual variability. And then this interindividual variability, which is the main subject of her research since 2020, when she's in Montpellier at the IPSIM, which is the Institute of Plant Sciences in Montpellier. Is this all correct?
Yeah, that's
good.
Right.
So very happy to host you here.
Thank you very much, and good morning, everyone. And thanks, Sam, for inviting me and for organizing these seminars. I think they are all always great. So today I will talk about interindividual variability, and I will start with the basics. Which is what we've been learning at school, that when you have one genome in one environment, then you will get one phenotype. And that's the genotype to phenotype link. And thanks to that relation, we were able to find the function of a lot of genes and their alleles and the phenotypes you get from that. But the reality is that this is an oversimplification. And what you will get in the real life that from one genome in one environment, you will have multiple phenotypes. Here I'm showing plants, but this is true for every organism that has been studied until now. And this is what I'm interested in, basically. And that's what I'm calling interindividual variability. So just to be sure that we are on the same page, I like to start with a bit of definition. So when I'm saying variability, I'm talking about the differences between genetically identical individuals or cells that are in the same environmental conditions. So it's not the same as the plasticity, which are changes in the phenotype in response to the environment. And it's also not the same as genetic variation, which are differences in the phenotype in response to change in the genetic sequence. Because in our case, we have no one neither the other. So what I also wanted to show as an introduction is that this variability, we were able to find some genetic context about that. So this is a paper in plants where they measured variability, interindividual variability for many phenotypes in different accessions of plants. So accessions of plants are different varieties of plants that have been collected in different parts of the world. And for each accession, they measured multiple plants and measured the variability for that. And here you have the distribution of the variability for the different accessions. So what you can see first is that there is some degree of variability, and it is changing from one accession to another. So it's not always the same level of variability that you will get in all accessions. And from one phenotype to another, you can see also that some phenotypes are more variable than others. And what is really interesting from this paper, and also a few other ones, is that they managed to do some quantitative genetics from that, and they managed to go to a region of the genome, and in one case even the actual allele that is associated with the variability. So it's showing that the variability that we are seeing here is at least partly controlled genetically. It's not something completely random. There is some control behind that. And because there is control, it means we can try to understand what is controlling it, and we might be able to ourselves engineer the level of variability that we want. And the other thing I wanted to show is, okay, we have this variability, it's controlled, but is it any use of having that variability for the individuals that have it, or the populations where it is present? And I'm showing you something that is known in unicellular organisms, where so you will have a population of cells, they are all genetically identical, but you will have two groups of cells, the blue cells that I would say are normal, and the red cells that express a gene involved in the response to stress, let's say antibiotic response resistance. And these red cells, they don't divide. But then the red cells can become blue at any moment, and the blue cells can become red at any moment. They are all genetically identical, just differences in the expression of that gene. Then, when you have a stress, let's say antibiotic treatment, only the red cells will survive and the blue cells will die. And then when you go back to an optimal condition, the red cells can become blue again, divide again, and recreate the original population. So you see here how having this interindividual variability before the stress is helping the population of cells to survive a stress that cannot be predicted, is sudden and strong. In plants, we don't have that kind of proof yet. We only have a strong suspicion that it might be useful in the context of variability for seed germination. In particular, for plants growing in the desert, there have been theories about that, but it has not been proven yet. So basically, when you have a rainfall, you will have seeds germinating, but not all the seeds germinate. Some will germinate later on or after the next rainfall, but they are still here on the soil. And if you have a drought, then the young plants are very sensitive to this drought and they will die, but you still have seeds in the soil. So then later on, they can germinate and allow the population to survive. So again, thanks to that, we suspect that this variability in seed germination might be useful for a population of plants to survive. And I'm still hoping that one day people will actually prove that this is true. Okay, so what I'm interested in is mainly what's happening at the molecular level. Because phenotypes are a bit complex to study, so I like to make my life easier. So I'm looking at gene expression. And basically, I'm looking at the transcriptional variability and trying to see also later on if it can lead to the phenotypic variability that we are looking at. And so during my postdoc at the Sensory Laboratory in Cambridge, when I started, there were a few examples here and there of a gene with variability between cells or between plants, but we didn't have a global view of the level of variability in plants. So what we decided to do is to define what is the level of variability that we have between plants, and if it can change during a day-night cycle. Because gene expression is changing through a day-night cycle, so why not variability also? So what I did was to do RNA-seq on single plants. So one plant here is one RNA-seq with 14 different plants in one time point every 2 hours through a day-night cycle. So that's quite a lot of data to harvest, to analyze. That's also quite a few nights I stayed in the lab because that's the experiment that worked. But before that, we had to optimize and so on. Which is why I'm not working anymore on circadian clock or time courses like that. I like to have my nights. So once we had this data, then the idea was to compare the variability between the time points. So inside one time point, what we can do is to compare the RNA-seq for the different plants and see what is the level of variability we have between plants for all the genes in our transcriptome. And then we can do that every single time point of the day-night cycle and see if the variability itself is changing through this day-night cycle. So I want to show a bit how we analyze the data, because at the time there were no tools on how to analyze variability in RNA-seq data. So basically, I tried many different tools, and I managed to hijack a tool that was existing so that it would fit my data. So what you have here for one time point is data for all the genes of the transcriptome. On the x-axis, you have the average expression for that gene. So when it's on the right, it's more expressed. And on the y-axis, you have the level of variability, which is here the square coefficient of variation. So it's the variance divided by the square average. And what you directly see is that there is a nice negative relation between the two. So we cannot have a simple way of analyzing the data and let's say, okay, let's keep all the genes with a square CV more than 0.1, and these ones are highly variable, because then you have a huge bias and you will enrich for the genes that have low level of expression. So what we did instead was to use that tool where you basically fit the trend for your data, and then you look at the genes that are far from the trend. And far from the trend is a statistical test, but like always, the statistical test, it depends on what threshold you want to put and so on and so on. But let's say that way we were happy to have genes that are far enough from the trend that we consider that they are more variable than the global genome, transcription for the global genome. So these, we consider them as the highly variable genes. And then to be able to compare them with something, we also took the least variable genes and some random genes, and then we basically wanted to characterize them. Do they have anything particular? Do we find any specificities in these genes? And so we looked at their function, at the genomic environment, and the chromatin environment. I'm not going to go through all the results because it's already published, already quite a few years now. But basically, this is a summary of what we found. And you can find the paper here. So the highly variable genes, we found that they were mainly involved in response to the environment. And that's also something that has been shown in other organisms before. So we were happy everything fit well with known data. But what was interesting, because we had this day-night cycle, is that we didn't have the same genes highly variable at the different moments of the day-night cycle. We had some genes that were highly variable, let's say only during the transition between the day and the night, or only during the transition between night and day, or only during the day, or only during the night. And they had different functions. So basically, we mainly have genes involved, let's say, in photosystem, photosynthesis in plants. These ones were highly variable just before the end of the day and just before the end of the night, for example. That's the kind of thing that we did find. And then other functions like response to cold and blood mainly variable during the day, and response to salt and iron mainly variable during the night. We also did find, and the lowly variable genes were mainly involved in basic processes, DNA, RNA, protein, metabolism. And then we also did find some specificities in terms of the environment of the gene. So basically they were smaller, but again, genes that respond to the environment are smaller. So then we don't know, is it because they are responding to the environment that they are smaller, and because they respond to the environment that they are highly variable, and then both are smaller? So again, that's the kind of thing that where you will need experimental data to actually know if it is because they are smaller than they are highly variable, or if it is just a correlation that means nothing. We did find that they were regulated by a high number of transcription factors compared to the lowly variable genes, and also that they had a compact chromatin environment compared to the lowly variable genes. And if there is any of you in the room or online who is working on plants, and in particular in Arabidopsis thaliana, which is a plant that I did use, I did generate an online website where you can look for any gene of interest and have the expression data for all the genes and the level of variability also for that gene through the day-night cycle, because it's quite a lot of data. So I thought at least it could be helpful also for the rest of the community and not just me having them. And I'm thinking of the people who are not coding and need this time too. Okay. So what I got from my postdoc basically is the starting of the hypothesis that I'm going through now since I got my position. And the different hypotheses and different things I want to look at is basically, the first question is the epigenetic environment regulating the level of transcriptional variability? A second one is, can we make a link between this transcriptional variability and phenotypes? Because for now, I'm only looking at expression of genes. If it stays at the level of expression of genes and has no impact of phenotype, it's not as interesting or as useful as if you actually have a link to a phenotype. And the last one is, can we see that because these genes are responding to the environment, can we see that the level of variability itself can be regulated by the environment? So that's the things I'm going to go through now. But before, I want to talk about the model I'm using. So I'm working on plants. And when I had a look at all these genes where we had a high level of variability, I realized that there is one pathway where we had quite a good enrichment of genes that are highly variable, and that the rest, that the nitrate nutrition plant. So I'm not expecting you to remember the name of all the genes, but basically here you will have all the transporters that are involved in taking in the nitrate from the soil inside the plant. And you can see that one of them has a high level of variability. It's NRT21. Then here are the genes that are regulating that transcription factor at the this transporter at transcriptional level, and you see that a lot of these are highly variable. And then again, we see quite a few genes that are highly variable at the level of assimilation of nitrate. And that was more than the global percentage of highly variable genes we had in the transcriptome. So I thought that might be quite interesting to look at. And also, the other thing that is quite useful is that we know a lot about this NRT21 gene. We know how it is transcriptionally regulated. We know how the protein is regulated. We know its function, and we have a lot of genetic tools to study it. So I thought that's a good model to start working on variability, because working on variability is quite uncharted territory in plants. So at least let's work on something where we have a lot of knowledge. And so the first thing I did was to check, based on the tool we had, if we can find again that this gene is highly variable. And the tool we are having in the lab is a luciferase reporter. So the luciferase reporter is a promoter reporter where you have the promoter of NRT21 fused with the luciferase. The luciferase is emitting light. And then when you have a camera where you can measure light, it gives you an idea of the level of activity of the promoter. So it's a way to know if the gene is highly, lowly expressed, and so on. And here what you have are plants grown on a solid media with the nutrients that it needs to grow, completely in vitro conditions. And we measure the luciferase signal, and you can see here the level of signal in the roots of the plants where the gene is expressed. And this is when the plants are in low nitrate conditions, when the gene is expressed and the protein has a function. And what you can already see by eye is that we have quite a few differences between the plants in the level of signal that you find. But we want to actually quantify that. So what we do is that we measure the signal in each plant, and then we measure the coefficient of variation, which is a standard deviation of the signal between the plants, divided by the average of the signal for the plants. And we do that for approximately 30 plants, which for now I'm suspecting is not enough. So I'm going to do some experiments by increasing that number to see what is the optimal number of plants we need to have a reproducible measure of the variability for that gene. So when you do that, you measure the CV for the NRT21 LUC reporter. We also have another reporter that we use as a control with a 35S promoter, which is a constitutively expressed promoter. So it's supposed to be not variable. So it's kind of our negative control. And you see here that we indeed have more variability for NRT21 than for the 35S promoter. So it's highly variable in our hands. So now I also wanted to show you that the level, what is this level of variability that I show when I say it's highly variable? So here's the actual raw data behind the CV. So you have the luciferase signal for plants. So every dot is one plant. For the plants that grow in low nitrate condition at 1 millimolar or high nitrate condition at 10 millimolar, in this condition, the gene is useful. In this one, the protein is not used at all by the plant. And you can see that it is less expressed here than here. And basically, you have a 1.7-fold difference between the two that we find in a quite a reproducible way. And then when you look at the difference between plants in one of these conditions, we are in the same range of difference. So we have the same range of difference between plants in one condition than between two conditions, one in which the gene has a use and one where it doesn't have a use. So for me, this was quite a good news because it means that these differences we have between plants in this condition, they actually can have an impact on the plant at a physiological level. So knowing that, we wanted to check using that gene as a model with the idea then to go genome-wide to go further to see if we have any phenotypic consequences of this variability. We wanted to know when the differences are established between plants, the differences in the expression, and try to look at the factors that are responsible for this variability. So to look at the phenotype I'm going to look at and show you is the actual function of the protein, which is to transport nitrate from the soil to the plant, and it's something we can measure. So what we can do is to use, so the nitrogen that you will have in the soil is 14N, and we can use 15N and feed it to the plant artificially, and then we can measure how much was taken in the plant, which we call that as the influx of nitrate. So this is what we did. So we measured first the luciferase signal on the plant, and then on the same plant, we measured the influx of nitrate, so the function of the protein. So we have the activity of the promoter and the function of the protein in the same plant. And what you have here, every point is a plant. The different colors are the different experiments that we've done independently. And you see that we have a very nice positive and quite strong correlation between the two. So basically, when the gene is more expressed, the protein also has a stronger function and is taking in more nitrate from the soil. And this is when the plants were first grown in conditions where the protein has a role. If we do the same thing in the condition where at 10 millimolar of nitrate, where the protein has no role, we completely lose the correlation. So it shows here that the correlation we are looking at is only visible when the protein actually has an impact and a role and a function, which is quite nice. So what we want to do now is to be able to actually modify the level of variability for the promoter and see if we change the level of variability for the promoter, ideally without changing the average expression for the promoter, can we also change the variability for the function of the protein? But for that, we need to understand how to modify that on the promoter, which we are not yet at that point. So the second thing I wanted to show was when the differences in expression are established between plants, because here I'm showing you data for plants that are 9, 10, 11 days old. So they are quite young, but still they have at least a few days of life. So when are the differences established between plants? Is it already from germination or is it appearing later on? And so to do that, what I'm doing is in collaboration with Hélène Javot, she has developed a system where we can grow the plants, and at the same time as they grow, you can measure the luciferase signal in the plants. So you can see them here growing. They grow through a day-night cycle, so in normal conditions. And you see the signal for all the plants. For some plants, we completely lose the signal, but you see that they also stop growing. It's because the plants that stop growing, they didn't manage to do a key transition in the life of plants, which is moving from the reserves of food from the seed to producing their own food from photosynthesis. And these ones, they didn't manage to make that transition, and so they stopped growing. At first, I thought that that was quite annoying because it meant we needed to have a lot of plants to be able to analyze a few. But in the end, it helped us to understand and know when that transition is happening and to know relatively to when we see the differences in expression happening, where one stands compared to the other. So in the end, it was useful. But it took me some time to realize it was useful. So from that, we can extract the signal over time for each plant, only the plant that goes through the transition, obviously. And you see here the graph for all the plants. So you see that at the beginning, they all behave in a relatively similar way. And then after one point, which I showed here with the purple dotted line, we see that for some plants, the signal continues to go up over time, but for others, the signal stagnates or even goes down over time. So this is when the differences are starting to appear between plants. And then we continued, so not in this condition because the system didn't allow to do that, but in other conditions, we did that in a way where we measured only once a day for 3 weeks. And when we do that for 3 weeks, we realized that over time, we maintain a very good correlation. So basically, the level of signal that the plants have at that level of their life is still correlated to the signal they have at 3 weeks later, or like here 2 weeks later. So it means that when you have a high expression type, you stay a high expression type. And when you are a low expression type, you stay a low expression type for the rest of your life, except a few rare cases when we change state. We did find a few plants that did move from one state to another. That was rare, but it did happen. And here, the gray line is when the transition from heterotrophy, so growing from the reserve from the seed, to autotrophy, so photosynthesis, is happening. So you can see that these differences in expression appear after that developmental transition. And I'm showing here, so the blue arrow and the purple arrow, the two types of plants that you have here. So if I run again the movie, you can actually look at these plants yourselves to be convinced of the data. So you can see at the beginning, no difference between the two. They are very similar. And from now, you see that this one has less signal and the signal goes down. And this one is very bright white, so very strong signal. And you see that they are still growing well, both of them. We have a few differences in terms of phenotype, but they are still relatively similar. I thought that was quite interesting. And if we do the same kind of movie, but for the 35S reporter line, then you can see that this reporter is behaving in the same way through the entire experiment. We don't have variability and we don't have these two groups of plants appearing. Okay, so now that we know that these differences, they appear around 7 days after germination, and then they are maintained for like several weeks, the idea was to try to understand what factors might be regulating it. So to do that, what we did was to take plants and do RNA-seq on single plants and look at the genes that are co-expressed with NRT21, thinking, okay, if some of the genes are co-expressed with NRT21, maybe they are the reason, or we can find between them factors that regulate NRT21. So, for example, here, if you look at the level of variability from this data, so the square CV against the expression for different genes, we have NRT21 that is here, so it's well in this area of highly variable genes. So everything is good in our data. And then I did put here the genes that are known to be involved in nitrate nutrition at one point or another, or that are known to regulate NRT21. And we find that some genes are also highly variable, and these genes here are transcription factors that regulate expression of NRT21. So maybe just these genes that are highly variable themselves, they are transcription factors, they are the cause of the highly variability for NRT21. So I was quite happy with this result. And then I thought, okay, let's have a look and see if each of these are co-expressed with NRT21 in the different plants that we have. And then when you start doing that, everything falls down. And basically, none of the transcription factors that are regulating normally or that are known to regulate NRT21 are co-expressed with that gene. None of them. So from what I see, it's not the variability for the transcription factor that can explain the variability for NRT21. The only thing that we did find co-expressed with NRT21 are other transporters. A gene here involved in nitrate assimilation, and the protein that are involved in the regulation of NRT21 protein itself, not the gene, but the protein. So it looks like what we are looking at is actually a transcriptional unit where you have NRT21 and these two genes that are co-expressed in the plants, regulated by the same thing, which we don't know yet what it is, and ensuring that then this is actually co-regulated at a protein level so that when the gene is more expressed, the protein is more active, because these two genes, they are necessary for the activation of the protein itself. So since we could not look at genes that we knew already had a role in NRT21 regulation, I thought, okay, let's start with an unbiased approach. And instead, what we did was to do a clustering approach where we clustered all the genes in the different seedlings. So every column here is a plant, and every row is a gene. And we got some genes that are in a cluster where we have NRT21, because it's not easy to see here. I did plot in another graph the expression in the different plants for NRT21 by just ordering the plants based on their level of expression for NRT21. And then you see that the expression in that cluster is relatively similar to NRT21. So priori, we have here an enrichment for genes that are co-expressed with NRT21. If we look at the functions that are enriched in this cluster, and you look at these, again, you will be surprised that you find nothing related to nitrate nutrition. So again, a new surprise. But instead, what we do find are things associated with photosynthesis on one side, and things more related to development on the other side. And when I found this related to photosynthesis, I thought, okay, that starts to make sense, because in plants, we know that the carbon that you get from the photosynthesis and the nitrogen you get from nitrate nutrition are both needed for the creation of amino acids. And so the two are coordinated in the plants. They are co-regulated. So maybe here what we are looking at is that between plants, we have differences in this level of photosynthesis that is associated with the level of nitrate nutrition, and the two are coordinated and different from one plant to another. To go more into detail, what I did was to take all the genes in the literature that are known to be involved in photosynthesis or in other steps of carbon assimilation and nitrogen nutrition. And then I looked at the correlation with NRT21. So that's what you have here. And basically, on the x-axis, it gives you the information of the type of relation. Is it positive or is it negative? And on the y-axis, you have the strength of the correlation. So basically, when you are above that line, it means you have a significant correlation. And you can see that we have half of the genes involved in photosynthesis that are correlated with NRT21, and almost all of them have a positive correlation. And this is more than the genes that are involved in nitrate nutrition. So it seems that it's more this co-regulation between photosynthesis and nitrate nutrition that is involved in this interindividual variability that we are looking at. So all of this brings us to a new hypothesis that we want to test in the years to come, where we think that, okay, at the beginning of the life of the plants, they are in this heterotrophic state. So the carbon source is coming from the seed, and then they do this transition where they become autotrophic. So the carbon source comes from the photosynthesis. And this is quite an important step in the life of the plants. And what we think is that for whatever reason, which we don't know yet, when they come out of that transition, some will have, let's say, shown in blue here, a higher metabolic state than others. They will have more photosynthesis, they will take in more nitrogen compared to others. And this is maintained through the life of the plants. And then because of that, because we have these different metabolic states, it's making that then the gene involved in nitrate import in the plant is starting to be expressed differently also between these plants. And we see these differences in expression that are appearing over time between the plants. So what I'm thinking is that this inter-individual variability that we find for NRT21 is the cause of that is something that is happening at this point of this heterotrophic to autotrophic transition. It's something that we need to test in the years to come. Okay, so that was the kind of physiological part of the project, and the data I've shown have been published at the end of last year. And now I want to show you unpublished results on a second aspect, which will be much shorter, which is to look at that chromatin. So I told you before that the genes that are highly variable, they have a more compact chromatin environment. And in particular, we did find that for a histone variant, H2AZ, which is another variant of H2A, we have a different kind of profile through the gene. So if you look at where H2AZ is present from the beginning to the end of the gene, for the highly variable genes shown in blue or the lowly variable genes shown in green, you have differences. So basically, the genes that are highly variable, you find H2AZ through the entire gene, while the genes that are lowly variable, you find H2AZ only at the beginning of the gene. And if you look at NRT21, it's also the case. So here is data from a chip-seq where you can see that you have signal for H2AZ through the entire gene, while for a lowly variable gene, you will have a signal only at the beginning of the gene. So from that, we ask the question, okay, is H2AZ having a role in that high level of variability? Can it be one of the reasons why a gene is highly variable? So to do that, we used a mutant ARP6 in which we are losing H2AZ genome-wide, and we put that mutant into our luciferase reporter line for NRT21. And then we looked at the level of variability. And so that's the result we got for many replicates for plants grown in low or high nitrate conditions. So you have the wild type and the ARP6 mutant, and you can see that in general, we have a reduction of the level of variability in the ARP6 mutant compared to the wild type. And this, whatever the condition in which the plants are growing, knowing that in these two conditions, the gene is regulated by different transcription factors. So whatever the regulatory network that is regulating that gene, we always find that we have a reduction of variability in the mutant. So from this, it seems that H2AZ is increasing the variability for NRT21. Okay, that's nice. But then do we find a link with H2AZ itself? So to do that, what we did was to do CHIP, where you use an antibody against H2AZ to get the region of the genome where H2AZ is present. And we did that in wild type and the ARP6 mutant. So this is the qPCR result of the chip. We are waiting for the sequencing result. I mean, it's here, but we have to analyze it basically. And so what you have here are data for a lowly variable gene, a highly variable gene, and NRT21. And I'm showing you the amount of signal we have at the beginning of the gene and in the gene body. So if you look just at the wild type to start with in the blue, you see that for the highly variable gene and NRT21, we have more signal than for the lowly variable gene, and in particular in the gene body, where we have no signal in the lowly variable gene. So it's what we were expecting from the data I had from my postdoc. So everything is good. And then what you see that in the ARP6, we have a reduction of H2AZ in these three genes. The thing is that for LRT21, we have a reduction of variability, but not for that gene in ARP6. So for now, we don't know if it's actually a direct effect, an indirect effect, or something else. So we are looking at genome-wide data to be able to actually answer that question. And so this brings me to the last experiment I wanted to show you, where we wanted to go beyond NRT21 and look at what is happening genome-wide. So what we did was to do transcriptomics, again, on single plants in the wild type and the ARP6 mutant, where we don't have H2AZ, and compare these two genes. Again, there are no tools existing to do this kind of analysis. So we discussed with a biostatistician to try to develop our own tool to do that. And so what we did was to start from the kind of representation we had before, where you have the average expression against the variation and that trend. And from that, what we can get is this difference between this deviation from the trend, which is a log ratio of the CV for that gene versus the CV that you would get at the red line. And this is showing you what is the difference between the variability of that gene and what is happening genome-wide. So basically, when you have a strong high value, it means it's highly variable. And when you have a strong negative value, it means it's really less variable than the global transcriptome. And it means that this way we can have a value that is independent of the level of expression, because genes are changing also their level of expression in the mutant. And so we can compare this in the mutant and the wild type. And that way we have this kind of distribution where you see how the variability is changing in the mutant compared to the wild type. So you can see that we have a distribution that is roughly centered around 0, more or less the same on both sides, so which means we don't have a global increase or a global reduction of variability in the mutant, and we are more looking at changes that are not global, but more on some genes and not just on one direction. So we have some genes here that have a reduction of variability and others that have an increase of variability. And we did put a threshold at 2.5 standard deviation to get these genes, which is a good statistical point, so that we know that we have things that are really different from the mean. And when you look at these in a plot like that, where you have the variability in the wild type versus the variability in the mutant, you see quite well that here are the genes that gain variability in the mutant. Here are genes that lose variability in the mutant. And if you look at the functions of these genes, we see that the ones that lose variability in the mutant, we mainly have genes involved in photosynthesis again. So we want to look at that more in detail. And if you look at the genes that gain variability, we have completely different function. It's genes involved in more in response to stress, things that are involved in response to pathogens, and so on. So we have very different type of functions. And what we are doing now basically is to compare this RNA-seq data with the chief-seq data that we have to try to discriminate between the genes where we might have an impact because of a change in H2AZ and more indirect effects of genes that don't have H2AZ but change variability because their regulator is changing variability. And that way we hope to find how H2AZ is involved in the regulation of NRT21, which seems to be something a bit complex. It's not just going one direction, and it's not affecting all the genes where you actually have H2AZ to start with. Okay, so with this, I wanted to thank all the people who've been involved in the work I showed. So from the work of my postdoc in the group of James Doc, 2 students work with me, Marcel Bataille and Zeynep Aydin. And since I'm at the ILSIM Institute in 2020, the people I showed the work of today are Alexandre Vetter, an engineer, and Charlotte Lécuyer, a PhD student, who've been involved in the 2 stories I've been showing. And thank you for your attention.
Thank you very much, Sandra. Perfect on time. So the floor is open for questions. Also, online, if you want to ask something.
I can
Break the ice.
Yes. I could ask you many things, but also I will hold myself. We have two things. One related to the issue you spoke about the inter-individual, but then the things that your system allows to measure a lot of intra-individual, which you have not developed. So how relevant is this? Because so given the level of intra-individual variation, how much can we learn of what happens in the individual by taking an average of it all?
Yeah. So I did look at intra-individual at a very at a cellular level. So I have another construct where you can actually measure the level of signal that you have in each cell. So we did that by having NRT21 in one color and the 35S in another color. And what we did find is that if you analyze only one cell type at a time, because we have differences in the level of expression between the cell types, then we find that NRT21 is not more variable between cells than the 35S. So it looks like what we are looking at least between cells, is something where you don't have inter-cell variability. It's quite homogeneous in that, but we have inter-individual variability. But then when you look at the movie, it's clear that you have regions of the root where the signal is not behaving the same way from one plant to another. And that's something we want to look at more in detail. For now, I'm just not sure how that's the problem, because then you're integrating temporal and spatial data at the same time. It's a bit complicated to integrate at the same time. What we did was to do things in a very crude way. Instead of taking the signal for the full root, we took only the tip of the root or only the top of the root. And then we did find that it didn't really change the global thing that is still highly variable, whatever the region you're looking at. So it's not like one region is not variable and another is variable. Both regions are variable between plants. But then we will need to go more in detail. And if anyone is really keen into spatial temporal analysis of imaging data, I'm up for collaborations, like really.
So I have a question regarding when you study noise, because to me it's always a bit scary that you think you have like an homogeneous environment, but there might be microenvironments within your environment, and also whenever you're measuring something, there's intrinsic noise in the tools that you're using to measure. So how do you deal with that to make sure that they are not impacting too much your measurements?
Yeah. So what we did was a very simple experiment. We got the plants growing in our petri dishes. We imaged the signal. We took them and moved them into a new petri dishes next to another plant, and we measured again after a few days, and perfect correlation between the two. So at least once the differences are established between plants, then the local environment is not affecting. Then I'm not removing the option that maybe the environment has an impact when the differences are established. But that's a bit more complicated, because then you need to really be able to control that at this specific time window, which is a bit more complicated to deal with. But once they are established, whatever you do on the environment doesn't matter.
We do have one question online from Ali Mouil. Unfortunately, her microphone is not working, so I'm going to read it. First, she said, thank you, it was super interesting. And the question is, she was wondering if the reporter misses part of the gene expression variation by not having the chromatin environment of the gene.
So it's a question we had, and the answer is that, in fact, it has the chromatin environment. So when we did cheap qPCR, we also looked at H2AZ in the luciferase gene itself. And we have the same luciferase on the luciferase gene than on the NRT21 endogenous gene. And it has also been shown for another chromatin mark in another paper before, when they also found that the reporter has H3K27 trimethylation, the same as the endogenous gene. And even the changes in response to the environment that they got from that chromatin mark is also the same on the reporter. So in that case, it seems that having the promoter itself is enough to recapitulate the chromatin, at least the very local chromatin environment of the gene itself.
So, thank you.
Thank you. So I understand why you are looking at gene expression for phenotype, but you have plans to look at more physical phenotype and even maybe going to fitness related phenotypes, and then to have influence on how it could influence selection, for example.
Yeah, so that's something I'm slowly moving on now. I have a PhD student working with me, but the data are still very early, so I don't want to show. But she's looking at phenotypes, actually. So many different phenotypes and how the variability of these phenotypes are responding to the environment. And so, for example, the kind of phenotypes she looked at are the architecture of the root, or the photosynthesis activity, because we have ways of measuring that. And what she did find is that in two different conditions, so she managed to find two different conditions where the level of variability for these phenotypes is changing. So the environment she's looking at is the impact of the CO2 in the atmosphere. So she's looking at the CO2 in the atmosphere as we have now, and as we would have at the end of the century, which is basically double of what we currently have. And when she puts the plant growing with double CO2 in the atmosphere, then she sees that for all of the phenotypes she measured, she has a reduction in the variability between plants, like for very different phenotypes, whether they are root, photosynthetic activity, or expression of genes, which was really surprising. Yeah. So now she's looking more at what are the factors that cause that reduction in variability. And the fitness part is something I'm currently considering for an ERC grant I'm writing. So where basically I want to see if we can get mutants where we have different level of phenotypic variability for phenotypes of interest, and then measure what is the impact of the amount of seeds you get, basically. And if this is important, where suddenly you get in stressful conditions where you have drought or stuff like that. But it's something that is quite a huge project, because what we are lacking now mainly is ways to change this level of variability in a kind of a controlled way. So that it's hard then to go into things where you want to look at the adaptative impact or stuff like that, because for now, if you look at different ecotypes, they have differences in level of variability, but they also have differences in the actual average of the phenotype you're measuring at. So they might be less or more variable for, let's say, photosynthesis, but they will also have a difference in the actual level of photosynthesis that they do. And for me to be able to show that it's actually variability, you need to be able to uncouple the two and to change the variability without changing the mean. Yeah, and that is something we don't have currently.
I think I have a question related to that and what was asked before, because now you say it's related to the mean. So what exactly, when you started in yours, you said like same environment, same genotype, same phenotype. So it cannot be same, cannot be zero variability. So what is that? Conceptually, what is that level of variability above which you will start to say, of course, you can do it statistically, but what is the level of variability that you can say really now things can be variable, given that there are constraints, maybe some phenotype or some activity, cannot change much anyway by default. I don't know, you cannot grow, I don't know, very tall. You can always grow very small. You see, or
yeah, see, yeah.
So how do you define that? How do you more high level? You say, okay, now this is more variability.
Yeah.
Really, it's more variability, or it's just measurement error, or just the normal variability.
Yeah, which is why I don't measure the variability itself. I always measure changes in variability. So either is my phenotype or the expression of my gene, its variability, is it changing in response to the environment? Is it changing this mutant? Is the level of variability for that gene different from that other gene, knowing they have same expression level? But I cannot say this is highly variable or this is lowly variable if I don't have a reference. It's not something I can do because like there is always technical variability in the way you measure. So you don't know if what you're looking at is technical or biological variability. So the only way I can do that is just to say, this is more variable than that for two things that you can compare, actually. So expression of two genes, the same phenotype in two conditions or two genotypes. So I cannot answer your question. And I don't think there is a way to answer your question to say really for sure, this is highly variable if you don't have a reference.
But then, if I may, if you change something, why you would not have a response? Why you would not have changes in variability if you change something? There's always be a response, right?
Not necessarily. So, for example, for the gene I just looked at, NRT21, and I showed today, when you change for example, the two graphs I showed you where you have low nitrate and high nitrate, and you see the change in level of expression, actually, we don't have a change in the level of variability between these two conditions. So if we take all the experiments we took together, we always have between 1.5 and 2-fold change in the mean expression, but we don't have a change in variability that is consistent and coherent. So sometimes in one experiment, it will look more variable or less variable in one condition compared to the other. But if you take everything together, it's not changing. So, and for that gene, actually, I have trouble to change this level of variability in response to the environment. Like I tried a different nitrate condition. I tried changing the sugar in the media. And the only condition where we got something is changing CO2 in the atmosphere. And these are things that are known to regulate expression of that gene, like very strongly. So, yeah. So I think you can change the mean without changing the variability. And there are studies showing that you can change the variability without changing the mean. So there are studies in unicellular organisms where they change the structure of the promoter and the binding site of the transcription factors, and they manage to keep the same average and change the level of variability for that gene. Mm-hmm.
So for the single cell, we show that when there's stress, only the red cell can survive, and then when there's without stress, then the red can turn to blue. So for the plants, show variations under different CO2 environment. And if you collect the seeds of the plant, and for next generation you change the environment to CO2 concentration, would the variation change go back to the normal?
That's a good question. We have not done that experiment in particular. What we did was to take the plants that had low or high expression for our gene and look at what is happening at the next generation. And then whatever the expression in the parent, we got the same distribution in all the next generation, in the descendants. So it's something that is, at least for that gene, is something that is re-established every generation, whatever you had in the parent. But I've not looked at something in response to the environment. Yeah.
Anything more online? Then, any last question? If not, then before we thank you, so that I will just repeat again that next week it's the Louis Dallaire seminar at the amphitheater at the at the Delegation with the Ment up the road, not here, at 11:30 with Annalisa Line from the University of Helsinki. And Sandra, thank you very much for your presentation.
Thank you for having me.